Sofian. The paper that we chose to analyze is interaction of APOBEC3A with DNA assessed by Atomic Force Microscopy. This was published in June 2014 in the review +1 by Tenko and his research team. We have a general overview of what we're going to talk about. To start with, we have a brief introduction of the APOBEC family because we had the conference last week by Jean-Pierre Bertagnon, so I guess all of you are up to date with that. And we talk about the uniqueness of APOBEC3A, why APOBEC3A in the family, and we have the biological question that these guys are going to answer. And after that, I'm going to briefly explain the concept of atomic force microscopy. Then we move ahead to the materials and methods, to the procedures that they've used in this presentation, in the research paper. And then my colleague Sofian will take over and talk to you about the data analysis, the results, and the discussions, and his conclusion. So to start with, we have a very brief introduction of the APOBEC family. As we all know, we have like 7 types of APOBEC3 proteins that are expressed in human beings, which are named A to G. So it's going to be A3A to A3G. And all of them play a role in inhibition of retroelements. We do know that too. And this is the enzymatic activity of all the APOBEC proteins. They elicit a single-strand DNA cytosine deaminase activity. Now this reaction over here is the one in which APOBEC proteins play a part in. You have the cytosine here, and you see the amine group that is deaminated, and you end up with the uracil. And this as Jean-Pierre Bertagnon said in the last conference, results in the types of mutations in VIF deficient or in HIV cell lines. The next we have the zinc coordinating domain, which has been structurally known to give you the catalytic activity of APOBEC3 proteins. We have either one zinc coordinating domain in A, C, and H, and the rest have 2 zinc coordinating domains. And all throughout these years, we've seen that A3F and A3G have been extensively studied because of their antiviral properties. Next, the uniqueness of A3A. Why at all should we study A3A in the first place? Okay, they form a part of the 7 proteins. That's a good reason to start with. But there are certain unique properties of A3A that make it so interesting. To start with, you have, it's not a strong inhibitor of the VIF deficient cell lines like A3G, because A3G is going to cause a lot of mutations in the viral cDNA, and A3A doesn't do that. And so the authors have postulated that it may have an antiviral role in myeloid lineages, not the T cells and the B cells, which are lymphoid lineages, but the myeloid lineages. And certain other studies have shown that it is involved in the clearance of naked double-stranded DNA. I think someone else is going to come and talk about that in due course of time. And we have the inhibition of retrotransposons in the human genome, which is also carried out by A3A. And the unique property of A3A is that it can deaminate both methyl cytosine and cytosine at the same time. But to deaminate methyl cytosine, it has got a 5 to 10-fold less efficiency when compared to the deamination of cytosine. So these are the 2 reactions. Cytosine deamination gives you an uracil. A 5-methyl cytosine deamination of that gives you thymine. And they've also shown that overexpression of A3A protein in the genome causes genomic DNA instability. Next up, the biological question of A3A. Now, what is that do we know? A lot of research has gone into knowing the biochemical properties of A3A as well as the structural aspects of A3A, but what we do not know is the biophysical properties of A3A. To start with, you have the DNA binding specificity. How specific is the A3A protein when it binds to DNA? And the stoichiometry in solution and in complex. Will the A3A protein exist as a monomer? Will it exist as a dimer or an oligomer when it is in solution, or when it is bound to DNA strands? And the last thing is the propensity to exist in a particular state. Does it prefer to exist as a monomer, or does it prefer to exist as a dimer or an oligomer? We'll see about that. So what the authors have done here is that to address these 3 questions, they have used a technique called as atomic force microscopy. Now this is a general setup of how an atomic force microscope would look like, and I take some time to explain how this works. So you see a cantilever here, you have a probe here, you have the sample, right? So this sample is going to move through, this probe is going to move through the sample. So let us say you have a sample, and this has got a small bunch over here, and your microscope, your probe, that is going to move on the surface, and it's going to have a peak over here. And here you see a laser that is focused on top of the cantilever, and it reflects back to a photodiode, right? So if my laser, at this point over here, if my laser is going to, say, deflect back to this point, when it moves over to the contour, there's going to be a change in angle of reflection. So there's going to be a shift of the signal that happens that is received by the photodiode. Now if the sample were to move sideways, which is now I explained to you how it goes this way, but then if it's supposed to go this way, like towards into the screen, say you have a frontal view of this. So there's going to be reflection, and when the point is when the probe is over here, there's going to be change in reflection. So there is in fact a deflection on this axis. So when it goes from there'll be a deflection on x to x dash and y to y dash as the thing moves. So when you place a sample over here, if you're going to scan it this way, you have a set of deflection on these two axes. If you're going to scan it the other way around, perpendicular axis, you're going to have a deflection on these two. So this signal is relayed back to a computer which assesses the whole thing and gives you a three-dimensional image of what the sample exactly looks like. And the image on the right is just a cartoon showing that the probe has nothing to do with how the sample looks like, but in fact, the laser beam that determines how the contour of the surface looks like. Now that we know how atomic force microscopy works, I'm going to run you through the brief experimental procedures that they have followed in this article. The first one is the preparation of APOBEC3 proteins. They prepare A3A, they prepare a mutated version of A3A in the catalytic domain, and they also prepare A3G. Now the reason why they prepared the C-terminal domain of A3G is a different thing altogether. I leave that to Sofian. And they prepared the hybrid DNA substrate, and we come to that. And incubation of the substrate of the protein that is produced, and lastly the sample preparation for AFM imaging. So to start with, we have the preparation of A3A protein. This is something that we all know we've done in the biochemistry lab last semester, sorry. So your A3A, which is tagged with a MIC and HIS, is cloned in a pcDNA 3.1 vectors, and it is transfected to 293 semi-confluent cells. It is expressed, it is lysed and extracted, and you have the lysate. The lysate is run through a column chromatography. You have a purified protein, and you assess the purity of the protein by chemistry blue staining and SDS-Page. And on the other side, you produce the A3G C-terminal domain. You do it by coupling it with GST in a PGX plasmid. It's very similar to the experiment that we did in the biochemistry lab. You put it into an E. coli, have it expressed, then you lyse it, then you have a glutathione resin purification, which is again the same thing that we did. And then there's an extra step here, which is called FPLC. FPLC stands for fast protein liquid chromatography. So in this step, what we do is that we have a liquid chromatography column. This is just to increase the purity of the protein that you had after your glutathione resin. So you end up with basically three proteins. The first protein is going to be A3A, the wild type. The second one is going to be A3A with a mutation of glutamic acid to alanine at the 72nd amino acid position. And then we also synthesized the C-terminal domain of A3G. Now that we have the proteins, we talk about the DNA substrate. Now, the DNA construct that they have is two double-stranded DNA flanking a single strand DNA piece. Now, the whole experiment is to find out whether A3As are going to bind to the double-stranded region, or to the single-stranded region, or to both. We'll see what happens. So the single-stranded DNA is 69 base pairs in length. The one on the left is 231 base pair. The one on the right is 441. So the whole experiment of atomic force microscopy is going to determine whether A3A binds to your single-stranded DNA, or to the double-stranded DNA, or to both. To the surface with deionized water, and then they dried it with argon gas. And then they used the AFM probes and saw what exactly happened. Is A3A bound to the single-stranded DNA or the double-stranded DNA? Next comes the data analysis, which my colleague Sofian will explain.
So the data analysis was done by a software called the femtoscan. Now I'm going to talk about the femtoscan for the simple reason that it was used to verify the position of the bond between the A3A APOBEC and the DNA protein and the DNA hybrid. The reason why is because femtoscan has the ability to calculate the length of the DNA hybrid used. Now, knowing the position of the single-stranded DNA inside the whole hybrid, we can actually calculate the distance between the binding site and then the ends of the hybrid DNA, therefore identifying if the protein linked itself to the single-stranded part or double-stranded part. The other thing about femtoscan software is that it is able to calculate certain dimensions of the protein, including the volume. The reason being is, the reason being is, forward.
Okay.
Yeah. The reason being is this is a standard image of AFM. Now, the only thing you need to know about the images in AFM is that this gradient here represents the altitude of the protein or the complex, the altitude compared to the surface. Now, knowing that the DNA is going to be on the surface of the mica. So if the protein links itself on top of it, it's going to be higher. And that is how they determine that these white blobs here, the higher points from the surface, are going to be the proteins. And I do say proteins and not protein because, again, they used femtoscan to determine the volume of these complexes, of these proteic complexes, to actually identify if the protein exists in a monomeric state, dimeric state, or just oligomeric state altogether. So the calculation is pretty easy. If you suppose that in a monomeric state, APOBEC's volume is 1 cube nanometer, and then what they calculate for the blob is 3 cube nanometers, then basically it's existing in a trimeric state. Now, they've done this hundreds of times with A3A wild type, and this is what they found. So they did a statistical analysis, and they came up with these histograms. The histograms are very self-explanatory. It's very easy. When they put A3A, they did the imaging with free A3A, which means A3A alone in the solution. They found that A3A had more tendency to exist in a monomeric state, which is seen here, obviously. And then they did the same procedure with A3A in presence of DNA. Then again, we see that the protein likes to be or prefers to be in a monomeric state, which means it retains its monomeric state after it binds itself to the DNA. Now, that being said, they can actually visualize the complexes between the DNA and the protein for very high protein to DNA ratios that are superior to 100. And this is a main difference between A3A and A3G, because A3G can do this in these experimental conditions for lesser ratios of protein to DNA. Now, I'm done talking about the wild type. This is where they moved on to the catalytic mutant A3A E72A. Catalytic mutant is basically a mutant that cannot exercise the biological function of A3A, which is a cytosine deamination function. But what they wanted to test out, if this mutation had any effect on the ability of A3A to bind itself to the DNA. And again, they do the same thing. They do AFM images. They see that the protein can bind itself to the DNA. By the way, the 3 here means that it exists in a trimeric state, 2 dimeric, and then we see that it can bind itself to single-stranded and double-stranded DNA. So they move on to the histograms, and this is what we see. This is the difference between the A3A catalytic mutant and the A3A wild type. We see that the A3A catalytic mutant is actually prefers more like to be in a monomeric state while by itself in a solution, whereas in contrast to A3A wild type, it actually has the potential of multimerizing when it's bound to the DNA. Again, these representations, these visualizations were only possible for high ratios of protein to DNA. Yeah, thanks, man. Now, this is where it got kind of interesting, is that they tried to compare A3A and A3G. If you remember what my colleague Venkat said, is that the difference between A3A and A3G in a structural level is that A3A only contains one single zinc coordinating domain, whereas A3G contains 2. So what they did is they basically expressed an A3G C-terminal domain, they express it, and then they did the same experiments, AFM imaging, and then the histograms, and this is what they found. For both A3A and A3G, they like to exist in a monomeric state, be it by themselves, when they are by themselves, which is, and we'll talk about that later in the conclusions. But, so, the conclusion, first thing they found, which they insisted a lot in the article, is that this was the first time there was a direct visualization of A3A being able to bind itself to double-stranded DNA, not only single-stranded DNA. Now the percentages were pretty asymmetric in a sense where 70% of the bonds happened between A3A and single-stranded DNA, and then A3A and double-stranded DNA only had a 20% complex yield. This led them to hypothesize that maybe this ability to link itself to double-stranded DNA allowed A3A to scan the DNA to find the single-stranded parts, which also, in a sense, they hypothesized that it allowed A3A to combat certain exogenous threats like papillomavirus, which can replicate its DNA in a double-stranded state. The second thing they found is that the mutation of A3A can bind itself with single-stranded DNA and double-stranded DNA, but when it binds itself to double-stranded DNA, it tends to oligomerize, forming mostly dimers, which also led them to believe or to hypothesize that maybe the 72nd amino acid, which is the glutamic acid, prevents or at least is involved in any kind of prevention of oligomerization. Now, the third thing they found is that A3A needs a high protein to DNA ratio, superior to 100, to bind itself to DNA, obviously in these experimental conditions. So they come up with another hypothesis, is that in physiological conditions, A3A would have a transient activity, meaning that it will bind itself to DNA for a short period of time, and in that short period of time, it will be able to conduct its own biological function, which they dubbed as the hit-and-run tactic. The fourth thing they proved is that A3G's C-terminal domain behaves in a manner similar to A3A's from a biophysical and structural standpoint, which led them to believe, again, or hypothesize, that the A3G's N-terminal domain is actually involved in the oligomerization of the A3G as a whole.
So that's about it.
And yeah, so that's about it. And we have the references.
Okay. Well done. Very